Hello everyone, welcome to my channel G5 Biology. This is Saroj Parazuli. Today we are going to talk about siRNA and miRNA, and we'll see what are the similarities and what are the difference between siRNA and miRNA. In short, small interfering RNA stands for siRNA and microRNA stands for miRNA. In the last video we talked about RNAi, RNA interference or gene silencing. We talked about the whole process of RNAi, and if you want to watch this video about RNAi or gene silencing, I'll put the link in the description section and you can watch this video from the link provided. And in this video today we are going to talk about the siRNA and miRNA, and we'll see the differences and similarities between these two types of siRNA and miRNA. RNAi or RNA interference is mainly done by siRNA and miRNA. So in the left hand side we can see this is the process of gene silencing by siRNA, and here on the right hand side this is the process of gene silencing by miRNA. And let's move ahead and see what are the differences and similarities between these two types of siRNA and miRNA. The first difference between siRNA and miRNA is in siRNA there is an experimental injection of siRNA, which is double-stranded RNA, or sometimes it can be a viral RNA that is affecting the plant or animal system. And similarly in the miRNA is the expression of an endogenous gene in the plant or animal cell. And the second difference is the presence of the protein called dicer. In siRNA there is a dicer that cleaves the double-stranded RNA into small pieces, and in miRNA there is a protein called DROSA. It cuts the miRNA into smaller pieces, and siRNA is cleaved into smaller pieces, and usually it ranges around 21 to 23 nucleotide long, while in miRNA the length of miRNA after cleavage will be around 18 to 25 nucleotide long. So this is going to be the third difference between siRNA and miRNA. And the fourth difference is the presence of Argonaut protein. So AGO2 or Argonaut 2 is present in siRNA silencing, while in miRNA silencing any type of Argonaut protein can do the silencing. And coming to the fifth difference, so fifth difference is the double-stranded molecule. So siRNA, so we see, so this is a siRNA and it will be a complementary double-stranded molecule. So this is siRNA. However, miRNA is a single-stranded molecule. So this is miRNA, and this forms the hairpin loop, and these all are single-stranded RNA molecule. So what happens when there are complementary bases? They pair to each other and they form heteroduplex system, while in siRNA there is a single duplex system. So this is going to be the fifth difference between siRNA and miRNA. And the sixth difference is the complete complementary base pairing. So what happens if this is siRNA single strand, then there is a complete base pairing between the mRNA and the siRNA single fragment. So the base pairing is going to be exact or perfect match. However, what happens in miRNA? In miRNA there is no perfect match. So the match will be usually in the 3' UTR region. So this complex is going to attach to the 3' UTR region, and if there is a random match, so if one nucleotide matches to this 3' UTR region, then the second nucleotide it may not match, and the third nucleotide can match to the 3' UTR region. So this is the sixth difference. So there is a complete perfect pairing between the siRNA and mRNA. However, there is no perfect pairing. Only there is the pairing of some nucleotide and some nucleotide may not pair. And this is the reason why we have the seventh difference. In the seventh difference, the single siRNA can affect a single gene. So what happens? One siRNA can target a single mRNA. However, due to incomplete base pairing, one miRNA molecule can target hundreds of mRNA. How it can do this one? So when there is an imperfect pairing, it can pair to the multiple mRNA region, and that is going to be in the 3' UTR region. So it can pair by imperfectly, and it can pair in multiple mRNAs. So it can target like hundreds of mRNA at a time. And the eighth difference is going to be the cleavage of the mRNA molecule. So in siRNA, what happens? This mechanism cleaves the mRNA into smaller pieces at the place of attachment. So it will cleave the mRNA. However, this doesn't happen in miRNA mechanism. In miRNA, this is attached to the 3' UTR region, and instead of chopping the mRNA into small pieces, it does the blocking of the protein synthesis. So it represses the expression of the gene, or it will repress the formation of amino acids. So that is going to be the eighth difference. And the ninth difference is going to be the chromatin remodeling. So it can only regulate the gene expression. However, siRNA can regulate the gene expression. It can modify the chromatin remodeling protein. It can do the histone modification, and it can also do the DNA methylation. And the 10th difference is going to be the organism where it is found. So this siRNA mechanism is usually present in the lower organism, plants and animals, and this miRNA mechanism is usually present in the higher organisms, higher plants and higher animals. So these are the main differences and similarities between those two, siRNA and miRNA. So let's summarize the similarities and differences between the siRNA and miRNA that we have recently talked. So it will be easier if I summarize in a table form. So this is a tabular form of the differences and similarities between siRNA and miRNA. There are 10 differences that we recently talked. So the first difference is going to be the experimental injection in siRNA, and miRNA is the synthesis from a gene. And siRNA is cleaved into small pieces by dicer, and in miRNA it is done by DROSA protein. siRNA is cleaved into 21 to 23 nucleotide long fragment, and miRNA is cleaved into 18 to 25 nucleotide long fragment. Fourth difference is the presence of Argonaut 2 protein, and in miRNA any Argonaut protein is present. Fifth difference is the presence of double-stranded RNA, and it is a single duplex system. However, microRNA is a single stranded RNA, and it is a heteroduplex system, as we clearly saw in a figure before. So this is how miRNA forms. So this is a heteroduplex system, and there is a perfect complementary base pairing to miRNA region, and in miRNA there is an imperfect base pairing. Therefore, it can target hundreds of mRNA at a time, and it targets in the 3' UTR region. And siRNA, it is complete base pairing, therefore it can only target a single transcript at a time. And the difference number 8 is siRNA chops the mRNA into smaller fragment at the site of complementary base pairing. However, miRNA, it doesn't chop the mRNA into small fragment. Instead, it will repress the translation by stopping the formation of the protein. And the ninth difference is going to be the gene regulation. And in siRNA, gene regulation, chromatin remodeling, histone modification, and DNA methylation, everything occurs. However, in miRNA, only gene regulation is seen. And the last difference is going to be the where it is present. So siRNA is present in the lower organism. However, miRNA mechanism is mostly present in the higher organism. So these are the similarities and difference between those two, siRNA and miRNA. In the next video, we'll talk about the practical applications of RNAi usually involving the siRNA. We'll mainly focus on siRNA approach, where we are injecting an experimental setup from outside and see how we can apply RNAi in crop improvement or as a gene therapy. Thank you so much for watching this video. And if you like my content and want to learn more about molecular biology, genetics, genomics, genetic engineering, and bioinformatics, please subscribe to my channel, like my content, and comment and share in your community. Thank you so much for watching this video, and I will see you in next video.